Hello, welcome everyone to the Masterclass on Fusarium wilt. For this session, we'll be talking about the TR4 incursion in Colombia, Australia, and Mozambique, and how to manage Fusarium wilt. My name is Anne Vézina. I'm the ProMusa coordinator. Before I introduce the experts who will be taking your questions, I would like to introduce the organizers of the meeting who made this possible is Guy Blome of the Alliance of Bioversity and CIAT. Hello, Guy. And
yeah, good morning.
And George Mahuku of IITA. Good morning, George.
Good morning.
And there's also Miguel Dita of the Alliance Bioversity-CIAT, but he hasn't arrived yet. But I want to thank him also for all his efforts for making this possible. So for our experts today on hand, I'd like to start with Rosie Godwin. She's the Research and Development Manager at the Australian Banana Growers Council, which is the national representative body for the Australian banana industry. She's been working in her current role for the last 5 years, working across all facets of R&D, but particularly TR4 and biosecurity. She's a plant molecular biologist by training and spent more than 25 years working as a scientist in universities and government agency. And we also have Altus Viljoen, who is a professor and vice chair of the Department of Plant Pathology at Stellenbosch University in South Africa. He works on Fusarium disease of banana, maize, and wheat, mainly fungal genetics and genomics, epidemiology, and the isolation of genes associated with the disease. He coordinates African Consortium on TR4. He established the African Society of Microtoxicology. He's a fellow of the African Academy of Science, and he's an international consultant on Fusarium wilt of banana and received several awards for his contribution on Fusarium wilt research. Tony Patisson is also with us. He's a nematologist and soil health expert at the Queensland Department of Agriculture and Fisheries. He has pioneered a range of technologies to assist in the quantification of microorganisms in the soil. His research centers around developing cropping practices for banana growers that suppress disease caused mainly by TR4, as well as managing nematodes to reduce the use of chemicals. Thank you. Welcome to you. Rosie, in Australia, in your presentation, you showed that after 5 years, you started with only 10 plants infected, and there's still only 93 plants that have tested positive so far, and it has spread only to 5 farms which are within 14 kilometers of each other. And you also mentioned that it's exceeded expectations at the beginning, but at the same time, there's been variable uptake of biosecurity measure on the farm. Can you give an estimation of the percentage of farm that have set up biosecurity measures, and what do the others say for not doing it after 5 years? What kind of reasons have you been able to see the barriers to?
Thanks. Yes, that's a very good question. I'd say that, well, there's a few questions there, but there has been variable uptake, and it's for many different reasons. A lot of times people have not uptaken it because they perceive that it's very expensive, and it really requires a systems change. So you've got the growers that are very proactive and who you know, that percentage of the growing community that will always be quick to adopt things. And I think lucky for us, a lot of those growers do live in the Tully Valley where those infested farms are. So I'd say in the Tully Valley, the uptake of on-farm biosecurity is very high. In some of our other areas, it's also high, but it's also variable, whereby some people put some types of biosecurity on their farms, all of the things that we would wish them to put on. You know, some people do have fencing and gates, and they might have footpaths and food exchanges, but maybe they don't have zoning of their sheds. So it's very hard to quantify, but I'd say overall, well, perhaps Tony could also have a guess at this, because I don't have exact figures, but I'd say maybe 20% of the growing community have high levels of on-farm biosecurity, and then, you know, it's variable lower down, and then there's also a small number that haven't done anything either, because they don't perceive that they are at risk because of their geographical location, or some people don't think they might have uncontrollable risks on their farm, so those people also perceive that it's not worth their while to make an investment if they can't control all the risks, say, because of flooding or something like that. So there's some of the reasons why it's variable, but we're still working with everyone all the time. It's, as I mentioned in my talk, and others have as well, it's still very important to us that all the growers that haven't got on-farm biosecurity do take it up, because there's so many farms that don't have TR4. I don't know, Tony, do you want to add anything to that?
The only thing I wanted to add, I totally agree, Rosie, was that it's a business decision. Many of the growers have made these assessments. They've attended the training that the Australian Banana Growers Council ran and the workshops. They're aware of the risks, and then it's a business decision with their situation and their farms on whether they can afford to implement biosecurity or not. So that's part of the reason why there hasn't been full uptake, that yes, some of them made this decision that at the moment the risk isn't high enough to them personally to invest in that money in that the money in that biosecurity. So the awareness is really high, but the adoption is probably a bit lower because of those individual business decisions.
There's no subsidies to help them.
No.
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 But is it offset by increase in productivity? Because I sort of heard some farmers who already set up the biosecurity measure that there is positive. Does it increase productivity in any way? Can it pay for itself? That's what I mean. Yeah.
Oh, I don't really think it could pay for itself, although some growers of our infested farms have noticed that, you know, they've got improvements in fruit quality, or just their business operations are more efficient because they've put systems in place. So, you know, they rolled those out on those farms that aren't even infested because they found it to be better.
Okay. Guy, do you want to ask a question?
Yeah, good morning, everybody. My question is for Tony. Tony, you explained that the activities that or the measures that need to be implemented vary according to, let's say, the number of affected mats or geographical spreads of the disease. And for example, maybe Australia was the only country able to keep numbers of mats affected to a very low number. I think you mentioned only 93. But I guess in most other places or other countries, numbers of mats or landscapes affected have kept on increasing in size. So now if you look at Mayotte, which is the latest or country or island to be affected by the disease, And as far as I know, only very few mats are currently infected. With your experience in Australia, what would be your advice for Mayotte, for the island, to do now to be able to stay at this onset of disease expansion?
My advice to the island of Mayotte would be listen to Altus and what he's got to offer as in their advice. I think he's in a much better situation. But I guess from what our experience, I was talking about management as the topic I was given. We've looked at the different scenarios growers face, and it really comes down to three different management aspects. Firstly, prevention, and implement everything you can do to prevent getting the disease. Then it's about management and stopping that spread of disease. So destruction of infected plants is at low infection rates is probably absolutely crucial. And I think within Australia, the farms that have implemented that most thoroughly and quickly are the ones that have had the greatest success in controlling the spread. And then, you know, resistance is a long way off. Is the third, management, once the disease becomes widespread. And I guess, yeah, to answer the question in my Mayotte situation would be to listen to Altus and to do what they're doing there, but it'd be to implement the biosecurity as much as possible, and destruction of those plants to stop the inoculum building up. But I guess in my times that I've been working with the disease, I think that's probably been since about 2005 now. I've seen a lot of cases in small farms and in different countries, and the disease doesn't always progress rapidly. It seems to be very much in situations of very intense production where the disease progresses very rapidly. In some of the smaller holdings, I think we see with all races of Panama disease, of Fusarium wilt, that where we have less intensive production, it doesn't necessarily progress. We still see Gros Michel grown in small holder farms in Central America. We see, in our case, Pisang Awak, which is very susceptible to race 1, grown in small holder plantations, and the same with Barangan and some of these other Gros Michel type in Indonesia, in smallholder diverse plantations. The disease doesn't progress as fast. So I'm not sure whether that answered the question.
Yeah, thanks.
Can I possibly add to what Tony has mentioned just now? You know, there are certain guidelines of dealing with incursions, and that's good to have. But honestly, it might get out of control very quickly. So the best thing to do is really to do a proper assessment of a situation with a new incursion, and certainly Mayotte is going to be different from what we found in Mozambique, and it's going to be different from what you find in Australia. So some situations might be much more vulnerable, if you can call it that way, and other systems might provide you with an opportunity to actually deal with the disease. So people should not just see this as a single recipe, you know, do this, do that, do this, do that with an incursion. You have to assess the situation. You have to look at the varieties involved. You have to look at the monoculture production, the density of this production, the history of maybe the incursion. Critical is to try and understand how this thing came into that specific area, because that can also help you to basically prepare for what might lie ahead of it. But one shouldn't see this as a one model fits all. kind of thing. It is really important that you get help as early as possible to try and deal with this. And I'm going to give you an example of, I think, very successful stories, and then other stories where incursions maybe have occurred. But if you look at India, where it came in the north, and we now know that it probably came in much earlier than when it was first seen. But that is, or it appears that the first cases were on commercial farms. I wouldn't say it's large-scale commercial farms, but it did spread, and it spread rapidly. And if you look then at the Australian situation, where they seem to have picked it up fairly early, they did a fantastic job. But then you need the resources and huge investment to do that kind of thing. And I think we have that kind of message from Rosie in her presentation. The Mozambican situation, they don't have that kind of money. It did come in onto commercial properties, but they are very few and very far away from each other. And we know now that it probably did enter Mozambique in around 2010. But the spread in the past 10 years has actually been incredibly limited. And this is simply because of the area, the whole layout of the banana production and how it's being produced in northern Mozambique. So it is important to be aware of these things. It also does allow us to prepare a little better for the rest of Africa. If you get that kind of a break, Australia had to act very rapidly, really, really rapidly to deal with the disease, and they did an excellent job, but few countries have the resources and the human capacity to do that as well as they did it.
Can I add on to your add on? I totally agree. But remember too, this is Australia's second time at it. We had the incursion in Northern Territory where we didn't do so well because we weren't prepared. We didn't know about the disease. We had learned from that situation that we needed to act quickly, but we then still had no idea when it first occurred just how widespread it was going to be. So this is why we had put in contingency plans. Well, first the biosecurity, and then while that was also being implemented, then the worst case scenario, we'd started investing in that, because at the early onset of the disease, we had no idea of just how widespread it was going to be.
Thank you. But Altus, before I get to the question in the chart, in Mozambique, that would be an example where the structured the NPPO was not as strong as in Australia, but the land tenure, because you mentioned in your presentation the fact that the land is public essentially, and then it was hard to stop movement. But did it play a major role in making it harder to control it, to contain it at the beginning?
Yeah, I think the difficult thing that happened with the spread originally was probably not because of local people moving through the land. They did walk through the plantation, but it's more the plantation workers that work there without any knowledge that the disease or the pathogen is there already. And, you know, talking to people afterwards, there were enough reason to actually investigate what was going on there, because plants got turned yellow and they were cut down and they were left there until it became just widespread and people realized this is a problem now. But it was the movement of people in land without knowing that the fungus is there, and they moved the fungus around. And then the second thing is obviously strong rains, flooding sometimes, which occur in most countries where bananas are grown from time to time, which spread the topsoil and the fungus to other areas. So there's a lot of things we can learn from that, just as Tony says, that how they learned from the Northern Territory. But really, I wouldn't say it was locals spreading it. But we now know it's the funniest things, like the feral pigs spreading it also possibly in Australia, you know, so one can never really predict what is going to move this thing.
Thank you. We have a question for Catherine Abadi about, are there studies on the quantitative TR4 inoculum, the dynamics during an outbreak incursion situation at the plot scale, how it's spread? I think that's the question. Does somebody want to take it?
I think Altus's information on the spread within the paddocks of the fields of Mozambique at Matanuska is probably the best at a plot scale. It's, yeah, it's not so good.
It's a complex question to answer, and Catherine can possibly just let us know if I did answer her question or not her question. But inoculum load does play a role in the development of the disease, no question about that. How it comes in originally and how that inoculum builds up is something that I do not think people have really studied properly. Maybe Stover many years ago did do some studies on that, but recently I do not know of that. What we do observe is that if you go back in the history, like the Matanuska case, and I can tell you India and some other places are more or less the same, is that people, as I said earlier, did observe yellowing of plants, cut it down, and then this thing goes almost quiet. You know, for a whole while, it can be 2 years or 3 years before it really comes back seriously. So what exactly happens in terms of the inoculum and the movement of this inoculum in that period, I do not know. The dynamics are complex, and I think studying these things are quite complex as well, simply because of the presence of that in the soil, which is not equal, you know. So you can study certain areas in the soil, but you cannot study it everywhere in that soil. Was it on, Catherine?
Can I add? We had a trial where we tried to a field experiment looking at rotation crops, which I presented in the video, where we looked at different plots after a rotation trial, and we used a molecular technique for the soil to try and quantify inoculum and how much was there. And we weren't able to do it because the variability within the plots themselves was so great that it just made no sense. The data was, well, it did make sense. There was no real change in the inoculum, but the variability within the plot was probably outweighing everything else. So it's very, very difficult to try and quantify it because there's a whole lot of other factors that come into symptom development, as well as the amount of inoculum in the soil. So it's, yeah, not something I'd rush out and do again.
Thank you. So we have another question from Salehuddin. It says he's come across a paper on lixiviate in Colombia coffee belt and its ability to reduce Ralstonia population by 30 in the soil by 32%. Has the substance been tested on Fusarium? Well, yeah. Anybody know about this coffee belt experiment?
I'll go.
Yes.
There was a recent webinar with from the Tropical Fruit Net, and the Chinese presenters, and I can't remember who talked about it, was certainly talking about a Lixivate in China that they were testing with for reduction in Fusarium wilt tropical race 4 in their soils. I just don't recall just how successful it was. I think they were saying that there was some reduction, but yeah, what the long-term effect was, I think, was still to be determined. So I think there has been some work out of China that they've been doing on this in this area, but I'm not fully aware of all the results from that.
Yes, thank you. On a similar line, Georges Mahuku is asking, we know the positive effects of increasing microorganism diversity in the soil as a strategy to manage FOC. Do we have information on the identity of microorganism, and what are the available strategies to increase their abundance in the soil? Tony again.
I'll take this one. Yeah, and that's a wonderful question, and I was hoping it would be asked. Actually, it's a lot of work that we've been doing in Australia with my colleagues from the University of Queensland, Dr. Paul Dennis and his PhD Henry Bird have been looking exactly at that as the banana microbiome, and so we've put a lot of work into trying to work out, well, what are the microorganisms that are most important to bananas, and they came across 36 bacteria which are found in every banana variety all around the world. So they're considered as the core of the banana microbiome. And also there are 21 fungal species, 21 fungal classifications that are also considered to be the core. They're found everywhere in bananas around the world, regardless of variety and regardless of soils. So they've compared those with overseas studies. So I guess our hypothesis is that if that's the core, then we get nice healthy plant growth when we get homeostasis, so we can maintain those microorganisms functioning within the banana plant. And there's different ones in the leaves compared to the roots, but the compartments are more or less the same. So we've got this core, and if we can maintain that homeostasis or even increase it, then we get this greater suppression of Fusarium wilt. We get greater suppression, or I guess, of the disease, of the expression of the symptoms. Where we get dysbiosis, where something has taken away some of some of the core, that's when we tend to get greater expression of the symptoms. So hot, dry weather, flooding, extreme cold, all these things tend to reduce the abundance of some groups of those microorganisms of that core. And as for their identity, the papers are about to be published early next year, so I'll leave that because I can't recall them offhand which ones they all were. But Fusarium as a whole is part of the microbiome of the core microbiome within bananas. So I guess that's not surprising. But what we also have been finding is that management has a big impact on what's there. So acid soils, low pH reduces the diversity and of bacteria and fungi, so that you don't have that same microbial activity that's occurring to support the core microbiome. High nitrogen applications very high nitrogen applications, regardless of the form, also have impact on that and tend to reduce the functioning of that core microbiome, so we don't get that diversity anymore, and we tend to see greater disease. So we're looking also at things like ground covers. How else can you do it? Ground covers is one way within Australia. In China, they're looking at biofertilizers and other amendments that can be able to increase it. So there's a range of different options there. So I don't know if I answered the question in the end, but yes, we do have a lot of information on the microorganisms, and we know who they are, and we're also finding out more about the strategies that we can implement to help boost those microorganisms. I can say things like bare soil, low pH, high nitrogen, which is probably common from many commercial banana farms, really favours Fusarium, and in opposite, doesn't favour diversity.
Yeah, and somebody's asking the question because they're in Colombia and they cannot do research on TR4. So whatever management strategies you find in microorganism, what's the likelihood, would they work regarding of the race of the pathogen, you think, generally, or is TR4 very specific or harder to control through microorganism in the soil?
Yeah, well, we can't work on TR4 within Australia, in Queensland, I should say, either. So we're working as part of international collaborations, and we're trying to validate the practices that we're testing as well on race 1 and subtropical race 4 in countries where tropical race 4 is a problem. And it comes back to what Altus said earlier, that inoculum level is a very important factor of it. So we've run some trials with ground covers under very high inoculum conditions, and they didn't work for a whole lot of other range of factors that we weren't able to, we didn't have full control of how the land was managed. And I think these other factors like high nitrogen, low soil pH were all, and very high inoculum, because we replanted plants straight after, well, plants dying around with TR4 and new tissue culture plants going in around them, which isn't the thing to do. Yeah, so the results weren't readily transferable, and I think that would be the case in most situations, that it depends really on the production system, but trying to get those key factors increased, can help the diversity.
Yes, Altus?
Karen, yeah, this is indeed really interesting research, which has almost revived in the past 5-6 years, where people have been looking at the microbiome, phytobiome, or whatever you want to call it, but in the soil situation around the roots, rhizobiome. To understand this better, and this is fantastic, making plants more resilient, I believe, but I wonder if Tony would agree with me that this should not be seen as a standalone way of managing the disease. One has to integrate the soil microbiome area with your other methods, you know. So if you have some partial resistance or something like that, or at least provide an opportunity for the plant to respond as well. I don't know, what's your opinion about that, Tony?
Fully agree, Altus. Yeah, none of this is meant to be a standalone treatment, that we build on it, and goes back to Rosie's presentation around the biosecurity, and just trying to reduce inoculum, preventing the plants. I guess what I'm talking about, there are a number of, a couple of different situations. I'm talking in the presentation mostly about very low infection rates. So we're trying to stop it from getting to that epidemic proportion. I think it's on one of your presentations, Altus, where we have an epidemic curve. I think Wayne showed it earlier, that we have a slow lag phase and then it takes off. So we're trying to extend that that slow lag phase for as long as possible. But once you become infected, heavily infected, and the rates tend to be over between 5 and 10%. Once you've got more than 5-10% infection rates, then it's gone. It's you're just waiting for the whole plantation to go down, and then you really need to look at cultivar replacements such as the GCT-CV 218s or other suitable resistant varieties cultivars. Yeah, so it's not a standalone treatment, and which brought me to the other point that I was meant to say. So what we're trying to do within Australia is we're looking at things that aren't immune, but if we can manage inoculum and we can manage the tolerance, it gives us a much greater choice of cultivars that we can choose from that have partial resistance or low level resistance. Because from our trial work that we've done with TR4 and selecting cultivars, finding resistance hasn't been the problem. We've started with resistant cultivars mainly out of the Taiwan program or readily available, but it's having them to be suitably agronomically suitable for the markets and not penalise the growers that grow them, which has been the greatest challenge. And I think we're starting to get on top of that now with our selections within that area. So they're not fully immune. If there's enough inoculum, they'll go down. But what we want to do then is have these other management aspects to help manage the inoculum, so that give these cultivars the greatest chance of continuous returns.
But what has been told in the last few days is that, yes, the GCT-CVs are an option when you're really in epidemic stage, that there's a danger that it will be masking the spread of TR4 and contributing to it because they're not immune. Well, I guess, if it's early in the incursion. If somebody, for example, in Australia wanted to plant GCT-CVs right now and they're not infected with, and they don't have TR4, or even if they have, probably if they have TR4 on their property, would they be allowed to?
Do you want me to take this, Rosie? Oh, yeah.
I think they can grow 218, can't they? Or Masana, they can grow.
Yep.
But no one would really choose to grow the one that we have in Australia at the moment because it has a longer cycling time and it's not as productive. So while there's so few plants, infested plants on growers' properties on the infested farms, they wouldn't really choose to grow one of those varieties, or their production is going to go down by a long way. It's just not, the cost of production in Australia is very high, so they want to grow the optimal variety as long as they can. And if you don't have TR4, you wouldn't think of growing one of those other varieties until you have to, essentially, is how I think we see it.
You can, just add on to what Rosie said. The work that we've done with GCT-CVs compared to Williams or 218, it's about 20% less productive. So you're getting a 20% production hit. We had some trial work in the Philippines where we were able to get a low inoculum. We were able to show that there was a penalty compared to Grand Nain, but if you've got the disease, then it's just a matter of time before Grand Nain will go down. So you then don't have that option. That it's the only thing you grow because Grand Nain is so susceptible. So there is a production, could be a production penalty, but if the disease is so high, then you have no other choice. And if everyone's infected, you're all in the same boat. But if it's only 1 or 2 farms, then you're really going to be behind. And I think that's for a lot of the varieties. So that's why we're trying to extend the plantation life for as long as possible.
Gordon, if I also, you know, again, like many things in Fusarium world, it's not a black and white kind of situation over here. One shouldn't just say, listen, Formusana is the answer or Formusana is failing. Straightforward. One has to look at the situation. Formusana is sensitive to environment. This is one thing that's really important. It's also very sensitive to inoculum load in the soil, right? So if you have very low inoculum, it does perform quite well. It stands up quite well. And I want to say there's often this thing of the new Formusana and the old Formusana, you know, the Formusana that was developed many, many years ago, and then the new one that's now available from TBRI. The partial resistance in this plant is probably the best that we have as an immediate thing. So if you have small growers, for instance, not severely infested fields, then, and they need to grow something, that is what is available. But there's a lot of work and a lot of research that is still required before we fully understand it. If we talk of 119, and Tony will know all these codes as well, GCTCV 119, that one has really good resistance in it, but its production is just not good enough for us at this stage. So if you can probably take that one through a mutation phase, I'm not sure if the Australians have included 119 in their mutation program. That's smart. That's the way that we need to develop these things and use what we have available at this stage. But one shouldn't just say, no, this is the answer or this is a very bad option. I'm going to tell you the C4, for instance, that's now being marketed by Rahan, is nothing different from 218, from Formusana. It's been a selection made in the Philippines, and this is actually what TBRI would recommend anyone bringing a Formusana into their countries, is to continue doing selections to find the clone that is producing best for them or working best for them in that. But this is where Tony's whole thing of the inoculum management comes in. You need a lower inoculum level in the soil if you want to plant Formusana fairly successfully. You need that. And this is where the integration of all these different options are coming together in the end.
Thank you. There's a question about the different, are you measuring, I think it's for you, Tony, the soil microbiome and the phytobiome? Are you looking at the microorganism within the roots?
Yeah, so the work that Paul and Henry did was they looked at 8 different compartments within the banana plant. So the bulk soil, the endo rhizosphere, with the apex of the root and at the base of the root, and then the ectorhizosphere at the base and the apex of the root, and then the endorhizosphere at the base and the end of the root, the corm, the pseudostem or the rhizome, and then the leaves. So they've looked at all the way. So compartments near one another. So the rhizome and the pseudostem, for example, have a closer, a similar microbiome than something like the bulk soil and the leaves. So as you move up the plant, the diversity decreases. So most of your diversity is in around that rhizosphere. What enters within the roots starts to decrease. As you move into the rhizome, it decreases further, the pseudostem further, and then I think there are only about 5 bacterial species from memory, bacterial groups in the leaves. So methanogenic bacteria were the biggest, most dominant group. So yeah, it's all been done, and it's all going to be published, I know, by the 31st of January, because that's the submission date for our Henry, the PhD student. So he's got to get it all out there.
Great, thank you. Rosie, there's been a lot of tracing that's been done in Australia, in Queensland, when at the TR4 incursion. Does it match? Well, I guess you haven't found the origin for the first farm, where it came from, or do you have ideas that you might be willing to talk about, or?
There are a lot of theories, but we still do not know whether the TR4 that we have in North Queensland came from the Northern Territory or whether it was brought in to Australia from elsewhere. So in terms of where it has come from on the farms, yes, it hasn't shown up anything yet. I don't know if Wayne would like to make a comment about the genetic testing at all, since he's been involved in doing a lot of the testing. Wayne?
Hi, Rosie. Yeah, well, I'm not a molecular biologist, but my understanding is that although there are some very small differences within TR4, that hasn't been of any use to determine a possible origin in this case. Like most isolates around the world are very similar. There are some differences, and you can perhaps track spread to some extent using that. Altus probably knows more about this than me, but in our case, there's been no definite result from that. I mean, a fresh introduction is just as likely as it is having come from the Northern Territory. Like we have backpackers as part of our workforce. There's TR4 in places like Bali, which are very popular with backpackers. So, unfortunately, with our global the global world we have today, an introduction is that easy. Someone could be in Bali one day and working on a banana farm in North Queensland the next day. And although we have very good biosecurity in Australia at our airports and things like that, I mean, things still do slip through from time to time.
Thank you. But Rosie, in terms of the subsequent, the farms on which it was found afterwards, it did match with the contact between the farms? Because if the fungus spread before the symptoms had appeared on the first farm, then the four other farms that found it, is it when you do contact, they have contact before?
Well, interestingly, they're all in very close proximity. And when when all the infested farms have been detected, that Biosecurity Queensland does a lot of tracing, finding linkages. And even at the beginning, they spent a huge amount of time and categorised the farms. We've got our surveillance strategy, which we review every year. But in the beginning, farms were categorised as determined by their linkages with machinery and huge numbers of other things, because some growers that are in the Tully Valley have farms in other places, etc., and they shared machinery. But ultimately, in 5 years, none of those linkages have showed up as, those farms that were outside Tully Valley have not become positive. And now we're doing a new surveillance program where we go back over every farm in the whole of North Queensland over the next 12 months. And it just seems to be that the 5 farms are all just so close to each other. So it's really, it's the proximity that is the highest linkage from what we can see. So in our new surveillance strategy, it's the farms in the Tully Valley now that will get the highest amount of surveillance on them. And then farms that are linked maybe via other means will get some surveillance, and that maybe, I think it's on infested farms, they're going to get surveillance, and I might need every 8 weeks, and then high risk farms maybe every 3 months, and then every farm in North Queensland will get surveyed once this year, just to make sure there's no hotspots coming up somewhere that, that are hard to find because there are ladyfingers growing up there and they could have race 1. So we just need to go over every farm again to make sure there's no hidden TR4 outbreak. But yes, it does seem to be proximity is the biggest linkage in Tully.
Great, thank you. So we have a question from George. Gene editing has been mentioned as a strategy with potential to develop tolerant or resistant bananas. What is the progress? Are there promising products from this strategy, and how soon can they be available to farmers? Yeah.
There's lots of work going on gene editing globally. I think James Dale, China, is definitely involved in that. UK is definitely involved in that, and then University of California and some other places are. So the technology works. It works in banana, but the thing is you have to edit the right genes, and that is the important issue that I think one has to, and I'm not talking about partial resistance. You really, if you edit these things, you would like to have complete resistance. So I think that might be the area where people still have to do a little bit of work, but the technology works for banana. It's just like GMOs. You have to find the right genes to target.
I think you also, the trouble is too with the difference in the regulations around the world. Like in Australia, gene edited bananas would be acceptable, even though GM bananas are not, and it's quite variable. So that also has an effect on how quick. I mean, these things may all be progressing at an experimental level, but in the end, they still have to be accepted by the regulators within the countries and then accepted by the general public. So, you know, we support research on all of these different areas so that hopefully one day we will be able to get a improved variety. But I agree with Altus that I just hope that when these varieties are made that it will actually have a resistance gene in it.
Can I just add on to that? It comes back to what also to what Altus said earlier is what's the demand? If you need something now, then Formosana 218 is the answer. If you need something in 2 years, well, some may be able, we'll have our mutagenesis and others will have something selected which is a little bit more suitable. But if growers have to wait for a GM or a gene edited banana, then we don't know how long that can take. Then they may go broke in the meantime because of the disease. So we have to develop other strategies as well. That's not saying that you shouldn't look at gene editing and GMO, but that's all part of the mix. Is we need some answers now for infected growers immediately, and have some long-term very resistant strategies such as the gene editing.
We have a question about the minimum inoculum that TR4 needed to establish an infection in Grand Nain. Do we know?
Well, I don't think there's any answers to that one. I think it's really a case-by-case type issue. I think there was a quote that you only needed 2 spores per gram for the race 1 infections in Central America. Was some work that I think Ken Pegg quoted in his epidemiology paper. But I think that really depends on your growing system and how you're growing the bananas, or yeah, a whole lot of other factors on what the disease expression, on whether the disease takes hold. But once it is there, it's there forever. That's I guess the one factor that can be sure of.
And I think we can learn a great deal of from the epidemic on Gromichel and the race 1. So you know, the epidemiology, the management, if it's race 1 on Grand Michel or if it's TR4 on, it's going to be the same. So we should also learn a little bit what happened many years ago. And I do remember very well that Stower made the statement at the time and said a few spores are enough to cause disease, to cause infection in a plant. Obviously, if you have a huge inoculum load, you're going to have a huge outbreak of the disease. But certainly there's not a lot, if those spores are viable, even if there are a few being introduced into a field, it may cause disease.
Mm-hmm.
Selvarajan, do you want to ask a question yourself, or do you want me to ask your question about chlamydospore? I saw that you
can ask, please. Yeah, you can ask that. Yeah, okay. Shall I ask?
Okay, so
okay.
Yeah, go ahead.
I shall ask. Yeah. See, everyone knows that in a field where chlamydospores used to be there for 30 years or more, even if you stop growing banana, then once again you grow Grand 9, a susceptible variety, then again it infects. And the reason is the presence of chlamydospore. So if we go for a Grand 9, just grow and make the chlamydospore to germinate and remove the plants and fumigate the field, then again if you go for Grand 9 cultivation, will the infection will reduce?
Altus? Oh,
yeah, it's a really nice idea, and I'm sure many of us have thought about this. And I do not think too many people have really studied this to see if you can kill it by getting it to germinate and then basically there's no banana or there's resistant banana in there. I do think you will have an effect there, but I'm also not sure if this germination tube or the hyphae is there and it doesn't find a product if it cannot produce another chlamydospore and go back into a dormant stage. So I cannot directly answer this question.
and I think from the work that we've been doing, looking at alternative hosts and looking at soils, that a lot of Fusarium just seems to tick over on alternative hosts, weeds and things, so that you always seem to have this reoccurring small, low-level inoculum that's continually renewing and viable in the soils, but you don't see it because it's not causing diseases within those alternative hosts like weeds and things. So we spent a fair bit of time trying to work out what could be hosts and what couldn't be hosts. And I guess what we're finding within some of that work was that that the fungus, or this is subtropical race 4, would form hyphal nets around the roots of alternative hosts and then start producing more chlamydospores, and then some colonization. But as if you killed the plant in any way or it desiccated, then the fungus just took over and produced masses of spore material on the decaying organic matter of the plant. So I think it's a nice concept, but as Altus had said, but I think there's so many other aspects to it of that, what keeps the inoculum present in the soil, that becomes very difficult to eliminate.
It's a really interesting topic to discuss, and I'm sure Wayne and also Miguel can add to this discussion. But if you, for instance, if you have a plantation that's severely affected, you take it out of production for a year or 2 years or 3 years, what is happening in those soils? And this is where you need to look at the survival of the chlamydospores in those soils, and do they survive as well on maybe alternative hosts as they can otherwise? Or even if you clean it completely, that land, how long do these spores survive? And as much as I can gather information from, there appears to, if you take the host out, it appears that you do get a drop in viability quite rapidly, quite soon, but you do not kill all the chlamydospores. So many of them will survive for a very long time afterwards. So, but viable inoculum will be reduced in that land, but not entirely. So it's a tricky thing. If you replant afterwards, there's a very good example, Rosie, up in the Northern Territory. At the time that Freckle came in, that they had to take out all bananas from that land. And that farm that's still in production, that's contaminated with TR4. Because I visited up there, and the grower is incredibly experienced with this disease. And we said, so what would happen now? And he said, well, the first year there will be a 5% incidence. The second year there will be a 20% incidence, and so forth. He could actually predict what's going to happen. But before that, they had a higher incidence even then. So it does help a little bit if you do break the cycle of continuous banana production. And I guess this is why the interesting research on planting other crops is coming into contention.
Yeah, I was going to bring that up too, Altus, that up on the commercial farm in the Northern Territory, they've had so much experience with the disease, and they had land that they didn't grow bananas on for quite a while, sometimes 5 years, and they've also planted, say, things like pineapples on it. But then as soon as they go back, and as you said, that's what they've decided to do, just grow those couple of cycles of bananas, and so they rotate around their farm now. So it's just a different style of production, I guess, and they are able to grow another crop that is suitable for their climate, and they're growing for a market that's up in the Northern Territory, so the locality's suitable for them. Whether other people can do that and what crops are available, I think needs a lot of more research, because over in Taiwan they've also done that sort of work with different rotating with rice and other crops that they grow there, but the TR4 does always come back. But yes, they might get a few seasons out of it first. That's, yeah, interesting.
But crop rotation also is used in China, and again, it's a different system. It's they don't own the land; they lease the land, and it seems to be easier in certain contexts to go in the crop rotation system. And it's not for export; it's for domestic consumption. For the export trade, it seems that it would really be a last resort. That it would, because it would have spread everywhere. Then it would change the whole industry completely. But seems unlikely to see it as a part of a solution anytime soon. Somebody also asked about avoiding the production of chlamydospore. Is there any research? Is that something also that could be looked at? They're really, really, really nasty. Are there any?
Can I just add on to that one, because there was the research that was done by Nolene Warman and Elizabeth Aitken, where they looked at the infection of using a GFP Fusarium. And it was once the plants senesced that you got that chlamydospore production. So the plant's infected within the roots within 10 days of inoculation. It could be found and then moved through the xylem tissue. But once the plant was entering senescence, the xylem tissue became clogged. Then it burst out of that xylem tissue into the rest of the plant. And as it senesced, then that's when you got most of the chlamydospore production done. So if you, well, the fungus is making most of those clematis-spores. So if you're able to get early detection and removal of those plants as soon as possible at low infections, then it may be possible to reduce the amount of clematis-spores that are returning to the soil, which is why in Australia we're using the urea fumigation and destruction of the plants. So we're not getting that, or trying to reduce the amount of clematis-spores that return to those soils.
Mm-hmm. Yeah, and it has to be done in a very specific way. And yeah, I think we talked about that. Yeah. Are there models to predict outbreak based on the spread in the Philippines? Has anyone studied how it spread in the Philippines? And
I think there's models that have been developed, but if you're talking at a field level or within a farm level, just going back to the farm that we were talking about before in the Northern Territory, I remember talking to the grower there, and they were trying to predict where the disease was going to turn up, and they said there was no rhyme or reason. It just, well, this is where external symptoms were going to be expressed, that they just couldn't do it. There'd be one plant in one corner, there'd be another plant down here, and then there'll be 5 over here. And I think we're finding very similar patterns on the farms within Australia with the low infection rates. Yeah, I'm not sure about the other. That's at a field level, at least, anyway.
But you did say in last year in the Australian magazine, Tony, that that bananas are challenging. You think you've worked it out all, but they don't follow the rules, and that seems to be the theme. It's like a never-ending puzzle.
Yeah, well, you've got bananas that don't follow the rules, and same with Fusarium. That's a difficult organism to work with.
Yeah, it stands out among well, the organism, but the plant also compared to other disease on other crops. It's more challenging.
And maybe also just to add this, I mean, this is what's making this disease a little bit more interesting than others, that it's so hard to predict really, you know, how it's going to spread, and it's just not airborne or working like many other diseases where you can actually predict the spread. And I, again, can give you examples, and Tony will know about this one particular farm in the Philippines that had been kept free of the disease while the neighbors were really severely contaminated for many, many, many years, neighboring farms. And then eventually it got contaminated due to irrigation from the river, where other people's, you know, inoculum might have landed in the water. So it's really hard to predict unless you have a system where there is no biosecurity present in an area. Then one can more or less predict, okay, the movement of people eventually this and this and this area, or even in terms of the irrigation systems, the water, all of those things, you can predict according to that. But if you have good biosecurity in place to protect yourself, I think you can actually keep your land clean for a long time. You cannot guarantee that, obviously, but it's not like a Sigatoka disease that you can predict and say, gosh, this thing is going to spread by wind, and the chance is very good that it's going to end up in this specific site. It's a little bit more interesting, I would say.
And talking, Altus, about biosecurity measure in South Africa, are farmers starting implementing them on their farms?
Biosecurity against theft is being implemented, so so that helps for the TR4 as well. But before that, no, they didn't. It's really to protect their crops against theft that many farms have been fenced in. which is a good positive development for us.
Okay. And you also mentioned black leaf streaking. Yesterday, I was reading it's a popular article. There's lots of articles on TR4, and it's always from the point of view of your view of how they should grow it banana. It's not based on knowledge of banana. It's what growers should do. If you're against monoculture, you're going to say the problem is monoculture. But in that article, they say that the spraying of fungicide in Latin America makes the bananas more easier prey for TR4. But there's no spraying of fungicide against black leaf streak in Australia, and it's still a problem. And there was not during the first epidemic. So does it create a sort of, if you're gonna, and the incursion in Colombia was on an organic farm because they're in drier areas, so they don't spray. Does it gonna encourage people to be in denial of those kind of talk of the problem without being alarmist? There's the other opposition, other extreme of just, oh, if you're organic, you're okay. No?
Yeah, I'll tackle this one. We'll start it anyway. There is that perception within Australian growers, some of the Australian growers, that they're organic, and so therefore they don't need to worry. And I think that is isn't true at all. If it gets in, then it still has a great chance of establishing it the same as anywhere else. I guess what I've been trying to do is look at how you then increase diversity, so that maybe that if it does get in, it doesn't go as fast. But there's nothing to guarantee that you won't be infected if you are organic. Yeah, so I think biosecurity should be regarded extremely highly on any organic farms or any low input type farms.
Yeah, I guess popular media and sometimes even fake news. There's some interesting stories out there on social media that you can find. What is important is really to try and look at the bigger picture over here. If you're going to have this thing introduced into your land, you're standing a very good chance to get damage. This is just it. If you have nematodes or not nematodes, if you have chemicals or not chemicals, this thing doesn't need anything of that. I've seen the damage it caused in Oman, for instance, where temperatures go up to 50 degrees Celsius, and this fungus is still happy to cause a lot of damage in that. I saw the damage being caused in China that goes down sometimes very, very cold conditions. It's just a nasty organism, and it doesn't need anything else to help us to cause damage.
No, go ahead.
It says you talk to me. You talk about the paradigm shift that should come in banana production system. So when we have a resistant variety someday in the future, what would be your suggestion to slow down the evolution of another such virulent race of Fusarium? Which I thought was a good question. And it's, I guess, something that, yeah, well, I'll put it to the other panel members, what their thoughts might be on if there's a resistant variety.
Variation is not occurring that often in the fungus. These clones are quite stable, and if you want to have pressure, then you have to see the wide scale that Cavendish is being grown in Latin America on race 1 and race 2 fields, and yet there's not widespread damage caused by that. So the fungus hasn't changed, even because of all that that pressure by the stable organism. I think the variation that we do find is mostly coming from Southeast Asia, and this variation is probably developed many years ago. We do find new VCGs, which is different from probably wide scale. It might be small mutations just in the VIC alleles. So it's a different mode of getting new VCGs into this. But there are also cases where we find very unique strains, really unique strains. And I think Randy Plutz has published on one of those, which is the VCG 01214. That occurs more or less exclusively in Zambia or Malawi. And there are other ones as well that I'm aware of. So these are the exciting or interesting ones, and one really do not know exactly what happened with their evolution. But I would not say it's a very recent event. If you get something quite unique as a strain of FOC.
And what's VCG? Somebody's asking a question about VCG 012425. Can it infect both race 1 and race 4? Oh.
Yeah, this is an interesting thing that I think we should investigate a little further, because sometimes there can be some confusion. But in terms of general, then VCG 124, 125 is not supposed to cause disease to Cavendish banana, unless there are some interesting events. An interesting exception is what is happening in India, that there were reports of 124 causing disease to Cavendish, and I think one should investigate those strains a little bit more carefully just to see exactly what happened in that instance. I saw the field, and certainly Cavendish got sick there, but I haven't isolated the strain from there. But the Indians did report that, and one has to accept what they have reported in literature. In terms of TR4, if that do infect all the susceptibles of race 1 and 2, there's also some conflicting reports, and one would like to see more in the scientific literature. But there are incidences where actually TR4 is not that damaging to some race 1 and 2 susceptibles, as was indicated before, that everything that's race 1 and 2 susceptible will be highly susceptible to TR4. That seems not to be the case entirely. So there is some, it just needs to get into literature that we can see more solid science behind this kind of thing.
Thank you. Thank you very much for a very interesting discussion. Thank you to the participants for your question, and to the experts for your answers. And this is the end of the masterclass. So thank you. And well, stay safe, everyone. Bye.
Thank you.